biosystems. To just give you a brief about what Christian Biosystem does, we have established in 2003. We are a biotech firm based in Mumbai. We focus on understanding our customers' project from a technical standpoint to provide cutting-edge solutions to support their research. We have a strong expertise and background in immunology, and we work with biopharma, CROs, vaccine, and academia at pan-India level. Today's webinar, we are with Integral Molecular. So Integral Molecular is based in Philadelphia. They are experienced provider for virology reagents and services. The support scientists working to understand and combat diverse viruses, which includes the seasonal flu, influenza, SARS-CoV-2, dengue, Zika, and many more. Their flagship product is reporter-virus particles, RVPs, and other reagents of Integral Molecular enable the scientists to study viral components without the risk of exposure to the live viruses or the spread of contagious illnesses. So today we have two special guest speakers, Dr. Christina and Ms. Megan Pitts, who will be giving you some insight on advancing antibody discovery and vaccine development with lipoparticles and the RVPs. A short brief about Dr. Christina. She earned a PhD in the biomedical sciences from Lewis Katz School of Medicine at Temple University, studying membrane protein interactions in T cell activation. Following a dissertation, she completed a research fellowship at the University of Pennsylvania in immunology and infectious diseases. She and her group at Integral Molecular specializes in developing the tools to accelerate the vaccine and the therapeutic development for current and emerging viral pathogens. Talking about Ms. Megan Pitts, she graduated from Drexel University and she has over 10 years of experience working in membrane protein expression and antibody discovery for preclinical and clinical applications. And her work has contributed to 4 peer-reviewed publications. At Integral Molecular, she supports customers at all stages of their projects, from understanding the unique goals to providing reagents and services. For today's webinar, I request all the participants to be on mute. If you have any questions during the presentations, you can drop the same in the chat box. We will have a separate Q&A sessions wherein your questions will be taken up. And now I would like to hand over to Dr. Christina. Thank you.
There we go. Is everyone seeing that okay? Thank you. Okay. So good afternoon, everyone. Thanks for joining. I'm Christina, and I'm the application scientist with the virology team here at Integral Molecular. My colleague Megan and I will be speaking to you about our reporter virus particle and lipoparticle platforms, a little bit about how they work and how they can help your research. And so I will be speaking first about our reporter virus particle platform before handing off to Megan. Again, a cursor. Move forward. Okay. So Integral Molecular is a biotech company located in Philadelphia, Pennsylvania. We've been around for a couple decades, and our foundation has really been in membrane protein expertise as well as virology expertise. And so we have over a couple decades now of experience manufacturing our lipoparticles. These were our first product that we began manufacturing. And in the time since, our products have been used by over 500 companies. And alongside with our partner projects, that includes companies such as AstraZeneca, Merck, Daiichi Sankyo, Ono, Context, and others. In our virology space alone, we have been working for many years on a government-funded study of viral envelope proteins. We now have over a decade of manufacturing our reporter virus particles, And in the virology space alone, our data and our products have been used in over 200 publications and patents. And we really got started in virology research. And so this was founded on biotechnologies for understanding HIV viral entry and through CCR5. Now we have developed virus-like particles for more than 7 viral families. And we've now also characterized over 850 viral envelope epitopes through these NIAID-funded studies. And so this is with the National Institutes of Health. And what I'm showing here is a selection of papers from over the years that show the variety of viral pathogens that we've been able to produce reagents for and be involved with for research. And so this begins with HIV, and then moving on, expanding into dengue, influenza, Ebola, chikungunya, Zika, and now more recently, SARS-CoV-2. And this is because of how versatile reagent our reporter virus particles are. And so what I'm showing here is how reporter virus particles have been able to be used in publications that span the entire development life cycle. And so reporter virus particles can be appropriate for studies as early as the discovery stage, the IND stage, as well as through phase 1 and phase 2 and 3 clinical studies. And I'll be talking about some of these papers a little bit later. And the main application of these reporter virus particles is in neutralization assays. And so neutralization of viral entry is a key correlate of protection from disease, which is to say that if we are developing a vaccine, for example, that neutralization of viral entry is a way to determine how much protection a population of patients will receive from getting a vaccination. And these are assays that are performed with sera or purified therapeutics. And so what happens is that when you have a reporter virus particle, if there's successful neutralization, such as with antibodies, this is able to block an infectious reporter virus particle from interacting with the host cell receptor and infecting the host cell. When this occurs, there is no reporter gene expression. If there is no neutralization, the reporter virus particle is able to bind with the receptor on the host cell surface and infect the cell. This leads to expression of the reporter gene. And so for most of our RVPs, it's going to be GFP or organelluciferase, and this will lead to production of a signal that can be detected in a fully quantitative manner. Finally, our reporter virus particles are safer to work with because they're single cycle, they're non-replicative. And so after this readout, you no longer have to worry about further viral particles being propagated. And so to tell you a little bit about the composition of these reporter virus particles, we use a couple of different core systems. The ones that are most commonly used are a lentivirus pseudotyped system, in which we have a reporter gene that is surrounded by a heterologous core, a lipid bilayer, and then the expression of an envelope protein for a virus of interest. We also have our flavivirus particles, which use own virus species proteins while still delivering a reporter gene surrounded by a lipid bilayer and then envelope proteins for those viruses of interest. And so because of this design, these reporter virus particles are safe to use in BSL-2. And so this is a way to also be able to study BSL-3 and BSL-4 level pathogens in a BSL-2 environment. It gives you much more flexibility of readout compared to live virus assays because now you have a choice of GFP and luciferase. You have access to increased throughput, and these are also highly automatable. And so these are compatible with 96-well and 384-well formats. And they're also much more technically simple to use than live virus. And so there are several advantages to using these reporter virus particles. And so with these reagents, you're able to use them for serum screening, antibody neutralization, and high throughput assays. A little bit about how they're produced. We have an ISO 9001 certified quality management system. And briefly, what happens in it is that we use plasmids to transfect into production cells. We're then able to harvest our reporter virus particles from supernatant. These get aliquoted and then frozen. And so after they are in your hands as the end user, these are able to be used in neutralization assays. And so what I'm showing here is an example of data from a neutralization assay. And so on the x-axis, we have increasing concentrations of a neutralizing or not antibody, and then signal, say, for example, GFP, to which we're able to normalize our percent of infected cells. What we could see, for example, is that if we have a control reagent either on the RVP or antibody side, we see that infection is maintained. And if we have a neutralizing antibody present, we're able to see a dose-dependent decrease in infection as the concentration of the antibody increases. And so because we're using a plasmid-based transfection platform, this allows us to have a fast response to new variants. And our ISO 9001 certified quality management system means that you can rely on very high lot-to-lot consistency driven by our standardized production protocols. And so this is something that now, with years of production experience, we've been able to ship to customers worldwide to be able to provide these reliable reagents. And we have put a lot of work into optimizing our protocols so that you're able to be successful all the way from our production of the reporter virus particles to read out in your hands. And so these are examples of protocols that we use in-house and that we share with our customers. They're the same protocols. And so what we found is that our customers really do a wide variety of experimental designs and assays with our reporter virus particles. And so what I have here are some of the considerations to keep in mind when designing some experiments with our reporter virus particles. This is everything from your experimenting plate design and deciding on a dilution range, from binding data for an antibody, for example, be able to plan out if a 96 or 3D4 well format is going to be best for your study, leaving room in your plate layouts for the appropriate control conditions or edge wells, which lie along the edge of a plate that are more prone to evaporation. Of course, it'll also be important to check your target cell health. And so this is beginning from an appropriate cell confluence and growth rates. Also, for cases where the target cell of a neutralization assay is a stable cell line that may be overexpressing a protein or receptor, sometimes the expression of those ectopic genes is maintained with a selection medium, so the addition of a selection agent such as zeosin. And so being aware of what the protocol is for those target cells before beginning the actual neutralization assay. And then in terms of readout, RVPs have been used in a lot of different ways. We've seen our customers use all of these. So being aware of what compensation controls, laser power balance for flow cytometry readouts, checking for complete cell lysis if you're doing a luciferase readout, and if you're doing microscopy, checking that you have an appropriate final cell density and therefore an appropriate initial cell seeding density. And so these are all components of things that will go towards a successful assay. So this is also supported by our optimized protocols. And beyond all that, if any of our customers has further questions about setting up the assay in their hands, our scientific team, which includes myself, are working to answer customer questions. And so what this has led to is an extensive catalog of RVPs for developing vaccines for a variety of viruses. And so SARS-CoV-2, as well as some other coronaviruses, are in our catalog. We have a lot of strains of influenza A and B, both seasonal and pandemic. Filoviruses such as Ebola and Marburg, alpha viruses, henipaviruses, and arenavirus, flaviviruses, which I'll also discuss some data from a little bit later. And then we also produce custom RVPs for our customers. So if there is a particular variant that is of interest to you, or another virus entirely, we have now years of experience developing RVPs for very different viruses, and we're happy to take those on to help you with those studies as well. And this is a catalog that we're continually updating and expanding. And so I will say that GFP reporter gene. RVPs for HIV and rabies virus are also coming soon. And so now what I would like to talk about are a few different examples of studies for some of our more commonly purchased RVPs. And so I'm starting with an example of data that was produced using our reported virus particles for SARS-CoV-2. And so here is an example, as I'm sure everyone is aware, SARS-CoV-2 has been a rapidly evolving pathogen. And so over the course of years, what we've seen are periods of time where there can be relatively few dominating strains, but also periods where there are a lot of emerging strains and a lot of variability within a patient cohort. And so what I'm showing here is a window of time that corresponds to when the study was published, and so ranging from 2021 to 2022. And during that time, we are keeping an eye on epidemiologic data in the literature to identify likely prototype variants. And so, of course, if you're developing vaccines for an evolving pathogen, you can't wait to have reagents to help you. They need to be ready to go. And so it's really valuable to have a partner that is also watching the data and thinking about what tools vaccine developers will need. And so because we have been watching the epidemiologic databases for years now, we have been developing RVPs for several variants of SARS-CoV-2 over the course of the pandemic, which means that now we have a catalog of both historic and emerging strains. And this is very valuable for characterizing breadth of neutralization or changes or loss of neutralization over time. And so what I'm showing here is a figure from a paper published on SARS-CoV-2. And what this group was able to do was they were able to measure neutralization across several of our SARS-CoV-2 RVPs. And so I haven't included all of them, but you can see a range of some of the strains that were included. And so this is all performed with RVPs. And what we can see is that in the study, because they were able to compare all these different variants, you're also able to capture the variability and neutralizing ability across time. And so our RVPs are part of a process that is really a partner to vaccine developers. Same goes for our catalog of influenza RVPs. And so we also, as I've mentioned, have several strains in our catalog. And so for this example, this meant that when a new strain of influenza emerged, we were ready to be able to mobilize our platform to rapidly produce a reagent for that new strain. And so what I'm showing here is a calendar and timeline of our work for when Atexus 2024 emerged. And so this was one that had been transmitted and then detected in dairy cattle in Texas before it was detected in humans. And so the human infection was reported on April 1st. And then after samples were collected and the sequencing data was published and made available, we then ran with it to develop plasmids and to optimize our conditions for RVP production, followed by all of our standard quality control protocols and shipping out to customers. And so in this case, because we have experience generating these reagents and we have experience optimizing and validating them, we were able to go from the date of sequence publication to shipping to customers in 2.5 weeks. And so not only were we able to produce reported virus particles, which are for use for microneutralization assays, and so this is with the GFP or luciferase readouts that I mentioned, we have also been developing a version of RVPs specifically for HAI assays. And so for people who are working on influenza, a lot of the assays that are important for validating and characterizing vaccine responses are going to be using hemagglutination and hemagglutination inhibition assays. We also have developed a reagent, titer safe, specific for that readout. And so with these two different products, we're able to, one, stay on top of the data to become aware of when a new strain is available, we're able to mobilize our experience and our processes to be able to rapidly get a reagent out the door. And this is all without sacrificing the production protocols and quality controls that we have in place. And so Integral Molecular is able to provide early access to reagents for new strains. Moving on to some of our flavivirus data, what I'm showing here is a paper that our company published showing neutralization data across the 4 serotypes of dengue. And so not just for dengue, but also for influenza, for SARS-CoV-2, when you have a variety of strains for a given viral pathogen, all of the risk and hassle logistics of being able to source and bank and maintain different serotypes or strains of a virus adds a lot of things that drain your resources to a study. And so in this case, for dengue, you're able to compare neutralization across 4 different serotypes. And so what I'm showing here is a neutralization assay again. And what we can see is that when we look at our negative control, so our non-neutralizing antibody in the white, across all 4 serotypes, we see that infection signal is maintained. There's no neutralization occurring. Conversely, if we look at our positive control, and so this is 4G2, which is going to neutralize across all 4 serotypes of dengue, we can see that across the serotypes, again, that we see neutralizing ability of that antibody. And finally, this is a great example of the specificity of our dengue RVPs. And so this is an antibody that will be specific for dengue 2. And so as we would expect to see, dengue 2 is neutralized, the other serotypes are not. And so this suggests that our dengue RVPs are faithfully capturing the surface of these different live virus serotypes of dengue. And finally, I want to talk about this case study. This was published by Takeda using our dengue RVPs as well. And this was to characterize their vaccine TAC-003. And so their interest in characterizing protection across the dengue serotypes was because the design of this vaccine is based on a dengue 2 backbone. And so even though envelope proteins from all 4 serotypes are being expressed by this vaccine, they wanted to see what protection was really specific for each of the other serotypes. And so what I'm showing here is neutralizing titers that are plotted. The way that they looked at neutralizing titers that were specific for the remaining serotypes 1, 3, and 4 were they took these sera samples from patients. This is across a few different clinical trials for this vaccine, and they depleted dengue 2 antibodies from these samples. And so the way they were able to do that was they used a live dengue 2 virus, and they used that to bind and deplete out dengue 2 specific and cross-reactive antibodies. After that, they were able to then use our RVPs to test what neutralization was possible, and that was specific for each of these other serotypes of dengue. And so here for each one, we can see our neutralizing titers across the mock-depleted samples. And so we can see that there's neutralization to a degree across the other serotypes. And then on the y-axis is what the neutralizing titers are after dengue 2 specific and cross-reactive neutralizing antibodies were depleted. And so what we can see is that there are neutralizing titers in many of these samples that are maintained for these other serotypes. And so this was published in the Journal of Infectious Disease and then also was included in their regulatory submission to EUMA. And this study was done actually with sera that was remaining from these clinical trials. And so the ability to use RVPs, which need a much smaller sample volume, meant that they were able to expand the questions that they were able to ask about the effects that the vaccine had on cross-protection across dengue serotypes. And so our RVPs are able to provide robust solutions for a variety of clinical applications and expand what we can learn from a clinical trial. And so I'll end my section before handing off to Megan by just summarizing that what the virology group is able to offer from intermolecular is reported virus particles for diverse viral families, and this is a catalog that's continuing to grow. Our critical reagents are the product of an ISO 9001 certified quality management system. And so this means that you're getting a very consistent, very reliable product. On top of that, we optimize our protocols and share these with our customers, and also beyond that, offer individual support to customers who are setting up assays in their hands. And this gives you early and fast access to reagents for new.
Thank you. So our lipoparticle technology is able to display the native structure of the given target membrane protein. How we are able to accomplish this is we are working with a parental cell line, and we're co-transfecting in a target membrane protein of interest along with our viral core protein. This causes an assembly and a budding event to occur, encapsulating the membrane protein of interest within the context of our lipoparticle, and we're able to then purify that particle out of solution for its use in downstream applications. And the advantages of using this technology is that we can incorporate complex membrane proteins, whether or not that's a GPCR, ion channel, or transporters, in their native conformation and presentation. Because these lipoparticles are budding off from the cell membrane, that conformation is maintained. Also, that target protein is also at high concentration. So we are able to report out an incorporation value in the terms of picomoles of target protein per milligram of total protein. And we're able to use that in terms of identifying what the concentration of that target membrane protein is within each of our lipoparticle preps. And then these can then be used in downstream applications, including immunizations in animals to elicit high immune responses against the target membrane protein of interest. And our lipoparticles can also be modified either by biotin or fluorescence for use in other applications as well. So these applications for lipoparticles span the array of antibody discovery and characterization processes. So like I mentioned, lipoparticles can be utilized in animal immunizations, specifically for antibody discovery to develop immune libraries. Immune libraries can then be utilized and how we use them at Integral Molecular in developing phage libraries and phage panning. Once lead molecules are identified from that phage panning process, they can be further characterized in either BLI or SPR assays. Lipoparticles can also be a source of antigen and ELISA assays as well. So our lipoparticle product can provide high quality native antigen for antibody development at many different stages. Next slide. So first I want to highlight and how we here at Integral use our lipoparticles in that they can enable high titer antibody responses in animals. So on the left-hand side, we are measuring titer responses against a protein of interest. And we were testing out different immunization processes. So on the leftmost side, we're testing the immune titer from animals with DNA-only immunizations. You can see we're not generating a specific immune response against the target of interest. However, when we are able to prime the animal in the form of DNA and give a protein boost in the form of lipoparticles, you can see that titer response is able to be generated to the specific membrane protein of interest. And that's because lipoparticles are able to maintain native epitopes from these membrane proteins. The target protein is present at high concentrations, and by utilizing a DNA and lipoparticle immunization, we can rescue titers that were otherwise not possible. I'm further showcasing this here on the right-hand side. We're able to test immune responses against a GPCR of interest You can see we're able to show a strong and specific immune response to the GPCR of interest over our negative controls. And in this cohort specifically, we're able to show 5 out of 6 animals showed high titer by flow cytometry against this GPCR of interest. From here, if you want to go to the next slide, the lipoparticles can then be utilized in a process called phage display and phage panning. So either immune libraries or even naive libraries that have not been created from immunized animals can display the fragments of an antibody, and that's showcased here in the different colored phage. These phage will take the antibody fragments and display them. Our lipoparticles can be utilized in a target binding format where that phage is able to directly interact with the target protein of interest and bind specifically to the lipoparticle. So anything that is displaying an antibody fragment that is specific to the membrane protein of interest is bound to the plate, and then you can wash off any unbound phage. From there, that plate can be taken and the phage that is specific to the membrane protein of interest can be eluted off. And the antibody fragments can be purified for further development and analysis, or the process can be done again. So this is an iterative process to find hits that are uniquely specific to the membrane protein of interest. And on the right-hand side, showcasing a customer data set where they were able to utilize our lipoparticles specifically to discover CXCR4 specific antibodies. So on the left, you can see that this is an ELISA signal. What the outputs were, and by round 4 they were able to find phage that had bound specifically to both our unmodified CXCR4 lipoparticles and our biotinylated CXCR4 lipoparticles. However, they were able to showcase that they did not have irrelevant phage that was specific to our null or CCR5 lipoparticles. So at the end of that, they were left with phage hits specific to CXCR4. Go to the next slide. When from phage panning, you can have a lead panel of candidates that you want to further characterize to measure the affinity for these antibodies against the specific membrane target of interest. And a popular assay to be able to do that is through biolayer interferometry, also known as BLI assays. A common instrument utilized for this is an octet system. And how this works is that there are streptavidin-encoded biosensor tips that are able to specifically capture the biotin that is present on our lipoparticle. And that is, you're able to load the lipoparticles onto the streptavidin tip. From there, we're able to reach a baseline of lipoparticle capture. That tip is dipped into a solution of antibodies, be able to measure antibody association in the form of an on rate, so how quickly those antibodies are able to bind to the target membrane protein, and then that tip is then dipped into a solution to measure antibody dissociation, so how those antibodies are coming off of the target of interest. And these two values, both the on and off rate, are able to calculate what the affinity of that antibody is against the target membrane protein. On the right-hand side, I want to highlight how we were able to utilize this here in a publication at Intermolecular. We were able to publish when we were researching the glucose transporter GLUT4, which is a 12 transmembrane protein. we were able to use our lipoparticles to express that GLUT4 membrane protein in our lipoparticles, coat them onto the streptavidin-coated tips, and actually be able to measure antibody affinity of our lead molecules that we had discovered against GLUT4. And by testing this, we were able to determine what the affinity of LMO48 was against GLUT4, along with a lead candidate panel that we had. Similar to BLI, lipoparticles can also be used in a surface plasmon resonance assay, or SPR assay, as well. So it's the same core principles as a BLI assay. However, the lipoparticles are coated onto the sensor chip instead of a tip. And by utilizing the on and off rate measurements, you're able to determine the antibody affinity. I'm highlighting that in the right-hand side here, where our customer was able to utilize a Biacore T200 and our CXCR4, which is as a GPCR containing lipoparticles, able to measure their lead panel antibody affinity as well. Next slide. And from this, our lipoparticles are a pure and stable reagent for your assays. So I've showcased here some different sets of data that highlight that our lipoparticles can be a source of the membrane protein of interest. So we know that lipoparticle preps are pure through the form of EM data showcasing that. On each prep of our lipoparticles, we perform dynamic light scattering analysis to determine the monodispersity of our preps, and we've shown time and time again that they are monodispersed samples. We're able to showcase that lipoparticles are stable over years, and this is a Western blot highlighting the stability of CXCR4 within the context of our lipoparticles, and that it maintains that purity over time. And then on the right-hand side, lipoparticles can also be a conformational source of these membrane proteins, since they are expressed within the context of the membrane. And we have repeated these ELISAs over time when lipoparticle preps are both kept at 4C or at -80. We have shown that our lipoparticles are pure and stable reagents over time for your needs in your assays. Next slide. And just like our RVP technology, we also have optimized protocols to support your needs as well. That really comes from our experience in over 20 years of manufacturing our lipoparticles. These are optimized and validated protocols. We use these internally here at Integral Molecular, but our customers have also been able to be successful as well. And you can really trust that you're producing quality data and accurate data in results for working with some of the most challenging membrane proteins. Next slide. And just to highlight some of our catalog offerings, we offer sample kit lipoparticles, which are our smaller scale kits. We offer these both unmodified and biotinylated for receptors CXCR4, CCR5, and CD20, and these are sold in 400 unit or about 50 to 100 microgram kits. Both the receptor containing lipoparticle and the receptor negative or null lipoparticle are also included in our sample kits. We have a suite of ready receptor catalog lipoparticles. So these are our membrane proteins that we have worked with. We have optimized their production conditions and can reliably and consistently produce these to order. And they encompass a wide array of different membrane proteins, including GPCRs, single, 4TM, transporter, and even some viral proteins as well. Next slide. And so just to summarize here, Integral Molecular's enabling technologies can really support your research in many different facets. So Christina went over our reporter virus technology and showcasing our pseudotyped virus particles with over 100 off-the-shelf solutions that encompass many different viral families. I highlighted our lipoparticle technology, which are virus-like particles containing high concentrations of these complex membrane proteins spanning from GPCRs, transporters, 4TM proteins, and many others. Next slide. So yeah, we just want to thank you for giving your time and allowing us to highlight our many enabling technologies here at Integral.
Okay, thank you, Megan, and thank you, Dr. Christina, for the wonderful presentation. Okay, so we have a few questions with you. Okay. So one of the questions from our participant is, can we have organelle-specific lipoparticles? And if you can guide in producing lipoparticles of the protein of interest.
Yeah, yes. So in terms of organelle-specific lipoparticles, because these lipoparticles are budding off from the cellular membrane, they are pinching off pieces of the membrane and not incorporating the specific organelles of the target cell. So it's only, you can think of it as a specific snapshot of the cellular membrane. And if there is a protein of interest, just like for our RVPs, we also offer custom protein production as well, if that is of interest. So if it's something that is not currently on our catalog, you can reach out and we can support custom projects.
Okay, thank you. I hope the question is answered. Any more questions? I request the participants to drop it on the chat box. Okay, okay, I think the question is answered. One more question to you, Megan. Can the lipoparticles be labeled or modified?
Yes, we offer a few different options for labeling and modification. So we are able to biotinylate our lipoparticles, and I showcase some data where that biotin is able to interact with specifically strep avidin, but we can also fluorescently modify our lipoparticles as well. And how we do that is we have a viral core protein that is tagged with our fluorophore of interest, either in the form of GFP or mCherry. So those are our two fluorescently modified lipoparticles that we can offer.
Okay, okay, thank you. To you, Dr. Christina, could you shed some light on how do you monitor and pick strains to develop the RVPs at Integral?
Yeah, thanks for the question. Especially for the faster evolving pathogens, and so SARS-CoV-2 and influenza, especially, these are the ones that we follow the most. And so there are a number of epidemiologic databases. This includes GISAID, for example, for SARS-CoV-2. And so, fortunately, not only are these public health databases available and updated with relatively little lag, but we're also able to look at what similarities new strains share with previous ones. We're able to look at changes in the rates of prevalence of each of these strains. And so those are all factored in to what strains we'll choose to develop next. But it's a pretty proactive process. And so generally our goal is to not only have reagents prepared for the strains that go on to become dominant, but to also have other alternatives. So we're ready for a few different directions that these pathogens can go in.
Okay, thank you. One more, once we order, the customer orders. Okay, one second, just one more chat box question we have. What are the supporting documents you would be providing when a customer is ordering an RVPs, or what are the series of QC you go through?
I'll answer, at least for RVPs. I think the process is somewhat similar for lipoparticles. So we offer a bunch more things. So there are the protocols that I described in the talk, but there's also several other ways that will support your customers. So for example, with a customer's first order of reporter-virus particles, we send along a freezing of target cells for those RVPs. And so what arrives in your hands are not only the reporter-virus particles, but also their target cells for infectivity assays. And then, alongside those protocols, we also send along technical data sheets because we are QCing infectivity for every single lot. And so this, because of this, this is also how we're able to recommend an input volume for infectivity, for example. And so customers are also getting specific information for their lot when they order. And then, of course, if there are other questions, the scientific team remains available for those as well.
Okay, so we have one more question to you. The participant want to know what would happen if RVPs enter the human system via respiratory passage or other means. So if we can elaborate on any study done at your end.
So we certainly have not used them for in vivo studies. They're non-replicative, so they're, you're not going to get propagation. Optimistically, depending on the cells that were accessible and depending on the RVP, I don't know if the question is if we would be able to measure signal from it. But certainly the end result of that would be that the reporter virus particles would not be able to propagate.
Okay, thank you. I hope the question was answered. Okay, so that's it. I think there is no more questions from the participants. Okay. So we'll just wrap. Thank you all for attending our webinar today. We are grateful for your participation and hope you found the session informative and engaging. We appreciate you taking the time out of your busy schedules to join us. Your engagement and thoughtful questions contributed greatly to the success of this webinar. A very special thank you to Dr. Christina and Ms. Megan Pitts for sharing their expertise and insights on lipoparticles and RVPs done by Integral Molecular. The recorded session will be sent to you, and also we request you to keep an eye out to our email on promotion offers, which is applicable only for the registered participants. Thank you once again. Thank you all.